So thank you very much, and thank you to the organizers. Yeah, it's been a very interesting range of talks. So my talk will very much be on the mathematical side. I do dynamics, I'm a mathematician, and I pretend to have that what I'm doing has some biological relevance. So it's about as generic a title as you can get. What I really want to emphasize is that this is a talk about modeling. All right, so I'm going to try to introduce what I think is an appropriate way to study regulatory networks. My feeling is that we've heard a lot of talks about ODEs. My feeling is that ODEs provide much too much precision for biology. The other extreme would be Boolean networks, and I feel like those don't provide enough. All right, so I'm going to try to find a way to sit right in the between. All right, so there's more here than probably should be tried to fit into a late afternoon talk. So please ask questions, and then you don't have to listen to so much. All right, so yeah, it's to your benefit to ask questions. So what I want to start with is a biological question. I'll talk then a bit about the modeling framework. I'll give you a part of the way that we organize the dynamics, that we understand the dynamics. I'll then use that to answer the biological question. And I'll go back a little bit more to characterizing the dynamics, because I grew up doing ODEs and dynamical systems, so it's not that I dislike it. So what I want to do is to convince you that what I'm doing can be easily interpreted in terms of differential equations. So that's part 5 and part 6. And then but I'm telling you, you shouldn't use differential equations. So then I'll come back and try to convince you that even without having written down a differential equation, I can tell you about dynamics in ways that at least people like me who grew up doing dynamics understand it. Okay, so that's the plan. So here's the biological question. You know, my idea of how cells separate is the mitotic cycle. The DNA separates, the nucleus separates, the cell separates, and now you end up with 2 cells. But apparently there are other things that happen. Endocycling is where the DNA divides, but the nucleus doesn't. And then it can keep repeating. And I am told that this is a picture of a budding yeast cell where this is the nucleus, and then you've got multiple copies of the DNA that have been replicated, all right? And my understanding is endocycling is actually reasonably common in a variety of cells. Okay, I'm working with Steve Haas, who's a yeast biologist at Duke. Whoops, went the wrong way there. And he's trying to understand a network that would describe both of these, and this is his proposed network, all right? And so, question as a mathematician that I'm trying to answer is, can this network explain both of these phenomena, right? So, let's start about the modeling, okay? And the most important thing about what I'm doing from my perspective is that I am going to explain all of dynamics for you, organize all of dynamics before I even start telling you what the dynamics is. Now, we have a 200-page manuscript that's on the archive at the moment, and I was only given 40 minutes, not 4 hours. So I'm going to try to explain these in the simplest possible way, and I think that's about as simple a network as I can put up, okay? So I have one node, and it self-regulates itself. I keep hitting the wrong button. Okay, associated to that node should be some quantity, and of course we're interested in understanding that quantity and the dynamics of that quantity. The assumptions I'm going to make, I'm telling you this is a modeling talk, so I'm going to try to be very clear about the assumptions. And I think of them as coming in 3 flavors. The first is what I would call the biophysical. So this is, the chemistry, the biochemistry that's going on. So I'm going to assume that, if this is a protein or mRNA, that it's got a decay part to it, and it's got a creation part to it. And if you want to talk about how these numbers are changing, it's fair for me to say decay plus creation results in total change. Remember, I'm trying to do all this without writing down a differential equation. That's why I put this in words. Now, of course, it's nice to have functional expressions. So, here's quantity, there's some kind of decay rate. It could be a function of elements in the network. And here's the creation rate. For explanation purposes, I need actually to be using this more complicated term to handle the network that I want to answer the biological question. But since I want to explain what's going on, let's just pretend that the decay rate is constant. Yes? Oh, yes. OK. If I had a big network, then F1 could be a function of a lot of variables. OK? Yeah, so I'm kind of going back and forth there. Of course, the whole point is I have no idea what this parameter is, because yeah, there are model organisms where this has been measured, but I'd like to be able to take this method and work on, arbitrary problems that biologists hand to me. I have no idea what F1 looks like, all right? But I can make, and this is where I'm thinking of the biophysical assumptions. This is supposed to be a quantity, so I'm OK with it being non-negative. This is supposed to be a decay, so that better be positive. And this is supposed to be a production rate, so that better be positive. And physically, there's some limit as to the rate at which you can produce things. OK, what do I think of as a biostatistical assumptions? Well, that's the network, all right? I gave you a simple network. The previous network was a little bit more complicated. But an arrow from the mth node to the nth node tells me that the quantity xm influences the production rate, because I made it simple here, influences the production rate of node n. And then the mathematical assumptions that I'm making is that, when I see a pointy arrow, I'm thinking that there's some kind of monotone increasing as a function of, SY5 influences SY4 in a monotone increasing way. The blunt arrow is monotone decreasing. And then at the end of the day, I would like to tell you that you could have been using ODEs, but of course no one knows the ODEs, and I don't want to talk about ODEs, but I want to have continuous dynamics as opposed to, for example, Boolean models, okay? So the dynamic, yes.
You assume that, how does it depend on itself? So
on the
itself. So you have arrows going.
Oh, none of these talk to themselves, I don't think, directly.
So your own production does not depend on your own.
No, not for the network that Steve Haas is proposing. For this one, it does. Okay, that is very problem specific. Is that okay? Other questions? Yes.
Yeah, is there any significance that some arrows point to these boxes and some arrows point to arrows?
So all these are post-transcriptional regulations, and these would be transcriptional regulations. Okay, so
how does it appear in the formalism?
In this term. Okay. Yes. Okay. Okay. But of course, I don't know what that is, and I really don't want to write that down. There is a place where I actually do something, and I'm going to hide it all from you, but I'm very happy to talk ad nauseum about that, okay? Okay, so what I told you is, from my perspective, the most important thing that I'm doing is I'm organizing all the dynamics before I even start talking about it. So what do I mean by that? Well, this is a one-dimensional system. It's not that I really dislike Boolean. I don't think Boolean does a good job modeling. There's lots of people that can argue with me. What do I mean by not doing a good job modeling? Is that it's not precise enough, but it's great if you want to compute, okay? So I want to keep track, I want to keep that. So here's my abstract phase space. And what is this? This is 3 vertices and 2 edges. Why did I draw it this way? Because the nodes of this edge are this, and the nodes of this edge are this and this, and it seems to me it's easier to communicate to you what I'm talking about than writing a list of 3 vertices, 2 edges, and then telling you which edges have which endpoints, okay? So it's completely abstract. There's no topology. This is just lists. One dimension because it's a one-dimensional problem. Okay, I am going to think about these labels as being special. This label right here is when there's no protein, and this is when there's lots and lots of protein. So let me start organizing things. I'm going to put a plus here. Why? Because if there's nothing here, then things should be growing. Over here, whoops, I keep doing this. Over here, well, if this is bounded, and so if you have a huge amount of the protein, the decay is going to win, all right? So what I claim is that this dynamics can be organized into 3 different types of dynamics. I'm going to make use now of this extra node, and this node is coming in because there's an edge there. So what am I going to do? Well, I haven't told you anything. So at this point, I can only make a guess. All right? So let's say that I'm at some value of x. I have no idea what it is. And I'm a little bit to the left of that. And I'm going to assume that my x is growing at that point. Well, what am I going to put here? I claim I have no choice. Why? Because f1 is a function of x1, but f1 is supposed to be increasing. That's what the pointy arrow says. So if this was plus, then this must be bigger than that, and so if I move to the right, this must still be bigger than that. Is that okay with everyone? Well, I took care of the plus case. The minus case actually gives us some options.
How can it be minus if it was plus on the left?
How can what be minus? The second one. This one? Yeah. I'm changing my guess. Okay, so let me go, can I go, so I said, let's just pretend that's plus.
You're saying it cannot be plus there if it was plus for a smaller value of x. If it's
okay, so this is the same, I claim there's basically 3 cases that I need to consider for this model. This is the first case. And the next case is going to be, here I made a guess of a plus, so now I'm going to make a guess of a minus. If I make a guess of a minus here, and I move and make it a little bit bigger, oh, this is increasing, so maybe it becomes a plus, or maybe it stays a minus. Is that, did I answer?
I don't understand how it can be minus if it
here?
is second one.
This one.
Yes, you go plus and then minus. If it's an increasing function, how can it go from plus to minus?
Because the other term is decreasing.
This term is decreasing.
I thought this was f.
No, this is this change that I'm trying to tell you. So this is really, really small. That's why I did it up here. Okay?
Because I'm not sure what you're plotting here.
So, yes, you're right. So this is plus. And the reason it's plus is because basically I'm at 0. So this is 0, but this is positive. Okay? So 0 plus positive is positive.
Depends on how f behaves at 0 again.
Okay, I made the assumption that it's the growth and that it's always positive. Okay, so that was my assumption. Okay? So this was trivially plus, and this is trivially minus because of the assumptions I made. Okay? I could weaken that, but it just makes everything messy.
So question?
Yes.
What if f1 grows more slowly than linearly? Is that a problem?
Yes, if it grows more,
Severe smooth or something?
I mean, then I'll just be seeing minuses, right?
Well, I was thinking of the plus plus.
Well,
because at the beginning, at 0, you have a positive derivative.
Oh, I see. Okay.
And then after that, yeah, you could go negative.
yeah. Okay. Okay. So these are the 3 possibilities. And I want to keep track of the fact that there are 3 possibilities. I'm going to call these things wall labelings, these plus and minuses. All right? This only came from the network and those assumptions I told you about.
Sorry, what was your theta? I mean, I don't know if we can see
It's a label.
Yeah.
It's going to be, you can associate it to a quantity of x. that if we actually want to do a particular problem, we probably want to do that. But at the moment, it's a label. And what I'm telling you is, think of it as some x value, completely unknown. To the left of it, I'm telling you, so if x1 is...
What I'm trying to say is clearly there's a steady state between the plus and the minus in the second case, right?
You're way ahead of me.
Okay. Because then it's not a steady state.
You're way ahead of me. I'm just trying to write down what are all the possible dynamics that I could have to worry about, but I've never told you what the dynamics is yet.
Okay.
Okay?
Well, it sort of gave us a formula.
It gives us a hint, but that's not the right thing to work with. Okay? I'm happy to take the questions. Okay?
I thought it was x dot was equal to that.
I'm not doing differential equations. There's no x dot.
It's a change, right?
It's a change, right? I know I'm being very pedantic, but I want to get, a philosophy across.
How do you know it doesn't change multiple times, minus, plus, minus, plus?
I made the assumption that f was monotone because of the arrow. I can do that, and I can...
Monotone, it can be this, and then this, and then, it doesn't have to be increasing at a constant rate. It can be increasing faster than x1, and it can be decreasing
Right.
slower than
So I actually can do that, but then I'd have to put in more thresholds. Yeah. But I can do that. Right? Yeah.
Why do you have the x1 coupled to the decay, but not to the increase?
It's just that I'm trying to pretend that I'm trying to convince you that this story that I'm giving you, I can actually do it for n-dimensional systems.
But why is it like a different form for the decay and for the growth? For the decay, we have x1 coupled to the gamma.
Because this is a decay rate. And remember, I could have put a g of x1 here, a g of x here.
You could have put the whole thing as f of x, right?
I could have, but I really am kind of walking through this by looking at the separation, right? Yes, that was one of my assumptions. That was the very first assumption that I could make. Decay plus growth.
And you also want, the point is, you want not, presumably you don't want things to become negative, so you need to have the x1 term in there. Anything you're going to do, you need that lockable term at type x1.
Right. Yeah, things should not be allowed to become negative quantities, right? Yeah. Okay. Did I answer your question?
That is kind of for convenience here.
What's that?
So it's kind of for convenience here.
Yes, it's
natural
convenient.
biology.
I see.
It's absolutely natural biology.
Yeah.
Otherwise, you will get negative solutions.
But you can also couple maybe x1 squared for f1.
Yeah, it could be. I haven't told you anything except I want it to be monotone. I do want it to be positive to keep the discussion going, because otherwise we're going to really run into minutia. All right. I don't claim to have solved that minutia, but we're going to run into minutia. Okay. All right. So there are three different I'm going to call these parameter nodes, because they kind of parameterize what kind of dynamics I can have. Let's be a little bit more, let me explain what I mean by parameterization a little bit better now. I'm telling you this is supposed to be some kind of monotone increasing function. This is a very nice monotone increasing function. Here's P0, here's P1, the low value, the high value. The only thing I really care about is that the low should be less than the high. Okay? Let me give you another label. And the label I'm going to give you is gamma 1, and now I'm calling theta 1, 1, because I really care that this was an edge that came from 1 back to itself. And I can associate to these three nodes these sets of inequalities. And I want to think about it two ways. This is just the labels, or it's very easy to go from the labels to numbers. If I think about labels and I want to go from this post-set ordering to that post-set ordering, there should be a place where this switches. And I'm going to keep track of that by putting edges between my nodes. Yes?
Sorry, what is this P1 and P0?
Well, if this is monotone, there should be some low value and there should be some high value.
The infinite soup of f.
Yes, infinite soup of f, for example. Okay? Right. But I want, this thing that I call a parameter graph, When I take a reasonable sized network, this thing's just going to blow up. So I don't even want to store real numbers because that would take too much memory. Okay? So these are just labels. Okay? And I'm just playing with the ordering of the labels. Okay? But it's very easy, I think most of you are probably doing it, to translate these labels into numbers. And I need that because at the end of the day, I would like to be able to convince you that I'm really talking, I can talk about differential equations if you care about that. Okay? Is that all right with everyone? All right. Now I want to model the dynamics. So now I'm going to try to answer Eduardo's question. I've got edges and I've got vertices. Vertices are low-dimensional things. I can't see a low-dimensional thing. So let me blow it up. So take every one of these, every face in this complex, blow it up to a top-dimensional cell so that everything in this case now becomes one-dimensional. If I have higher dimensional complexes, I'm going to blow it up to higher dimensional cells. And I'm now going to build, most people would probably be familiar with the notion of state transition graphs. So now I'm getting back into the Boolean side of things. But I'm going to write it down this way. Cell goes to bunches of cells because this kind of looks like a map which I can iterate to do dynamics. All right? So it's just notational. It's a directed graph or a combinatorial multivalued map. Okay, so here are my wall labelings. The wall labelings come from the network that I gave you. I blow everything up, and now I want to tell you what the dynamics should be. So this, and here I'm using the fact that I'm going to work with an ODE. So if I have a cell and I have a continuous process, I should just move to my neighbors or stay where I am. This guy is moving to the right. This moves to the left. This moves to the right. Monotonicity says, well, if the left side is moving to the right and the right side is moving to the right, eventually the whole thing moves to the right, so don't bother to put a self-edge there. Over here, that's the monotonicity. Over here, the same argument, but now I got something from the left coming in, I got something from the right coming in. I'm going to get stuck, and so I should put a self-edge.
Well, how do you know that the going to the right may not approach infinite time, I mean infinite times and asymptotic limit, finite limit?
Because here I'm telling you that I'm moving, I'm telling you the rate of change is positive.
Strictly positive.
Strictly positive.
Sorry, it is a lower bound. Sorry.
Yes.
It was a lower bound.
Yes.
Sorry.
Okay. Okay, so you fill the rest in. There's some, on one dimensions, you can't go wrong. When you get to higher dimensions, there becomes an issue. I'll come back to that later. Just pretend that we can all do that, okay? So here are my state transition graphs. Okay, so let's characterize the dynamics. I gave you the model for the dynamics. I'd like now to characterize it. And what I'm going to do is I'm going to say, if I care about dynamics, I care about recurrence versus gradient-like dynamics. If you look at this, everything moves to the right. Oh, here it goes back to itself. It goes to the left. It goes back to itself. What happens? Well, I end up someplace. Over here, oh, this is interesting. I can go to the right and left. I can go to the left and right. So this is what I end up with. And you see, when you leave this, you end up down here. All right? And likewise, down here, everything goes to the left. This Morse graph, well, it's a poset because it's telling me this might be where you have recurrence. And if you're not recurring, I'm telling you the direction in which you move. Okay? Let me give a little bit of foreshadowing at the moment. Why do I care about the fact that I've already organized all the possible dynamics? Because we want to understand, I think, what happens as you move through parameter space. So, right, this is my parameter graph. For each node in the parameter graph, I've told you something about the dynamics. If you walk through the parameter graph, you can very easily identify cells. And so, well, this cell is identified with that cell. And so if you think, oh, now let's just pass the real numbers. So I'm in a parameter space here, and I have a continuous path. Well, there's got to be something here, and there's got to be something here, so there should be something all the way in between. And so there should be kind of a path of, for lack of a better word, fixed points. And here should be a path of fixed points. Let me just kind of draw another path of these fixed points. And this really looks kind of like a hysteresis diagram. Is that?
So I still don't understand, if you're willing to have one extra state that arises from, in some language, a Salomon bifurcation, why couldn't you have 5 things there? Why can't you have 7 things there?
I could. I'll come back to that. I'll come back to that at the end. Really important point. Very important point. Yes?
What do the colors mean? I can see that there's 3 different things in the second row.
I just want to keep track of the fact that this cell came from, that these cells are the same. That's all I'm trying to keep track of. Yeah.
So what's the point of the blow up? Like, for instance, all this picture couldn't we have seen before from all
You could have, but I want to tell you that at the end of the day that I have theorems about differential equations, and I want to use algebraic topology. And I know fixed point theorems that will let me say, oh, there's got to be a fixed point in here because you have an interval that maps to itself. You have to have a fixed point in here because you have an interval that maps out of itself. All right, so this is the setup so I can do algebraic topology. Okay? So I can think of it as a combinatorial hysteresis diagram. A natural parameter would be decay rate. All right, and what I would like to do by the end of the talk, but I might not get there, which is okay, I'd like to tell you, yeah, I know how to match this kind of combinatorial structure to the continuous dynamics, and that there is this really good model is good enough unless you absolutely tell me that you've got data that says I have to do something more complicated. Okay, and I'll try to justify that. So, you know, these are regions in parameter space, regions in parameter space. It's on the edges that bifurcations are taking place. I have kind of a dual complex going on here, if that means anything to anybody. Okay, what happens in general? In general, you have a high-dimensional, you know, big graph, and I'd like to know the recurrence and the non-recurrence. I'm doing topological sort, and I'm looking at sub-pieces of the topological sort that correspond to recurrence. Okay, it can be done really fast. That's an important point. And at the end of the day, the Haas diagram of the restricted stuff is what I'm calling the Morse graph. What I care about is when I show you a Morse graph, recurrence is associated with nodes, gradient dynamics is associated with edges. And I claim at this point I've given you enough information so we can go and solve the biological problem. Okay. Steve has proposed this network. Is this a reasonable model? Okay, here's wild type. This is time series of expression levels of these genes. I think it's mRNA, but that's getting beyond my pay grade. How am I going to deal with this? I'm first going to identify the parameter graph associated with this network. How many nodes in that parameter graph? Because I'm not going to draw it for you for obvious reasons. Right? You know, there are 51 trillion something nodes. And that's because when I do the parameter graphs for this thing, I'm telling you about the dynamics of that network and every subnetwork. So what I'm going to do is I'm going to restrict to essential vertices in my parameter graph. And that is going to be only those parameters where every node and every vertex actually has something to say with respect to the dynamics. So if I go back to here, okay, what do I have to do? Oops, one more. In this dynamics, everything goes to the right. It doesn't really matter what x is. Over here, x actually plays a role. So this would be essential, and these are inessential. Okay? That's really all that's going on. Okay, and we can identify that. Okay, so what I'm really doing is saying I'm not going to look at the 51 trillion regions. I'm only going to look at a billion of them. But and the reason I put this up is not because I do want to show off the big numbers, but if you're doing an ODE model of this, and you've just got a whole bunch of parameters, I don't know, maybe 50 or 80 dimensional parameter space, and you don't know what those parameters are, and you just start randomly sampling, how are you going to sample? I'm telling you, you better start with 51 trillion something samples, and that's not even going to tell you the global dynamics, all right? So I'm telling you how complicated these problems are. No offense to anyone, because I do appreciate that models speak to problems, right? But I would say people who are doing sampling, random sampling, are being incredibly naive. Because even this relatively simple ODE is requiring, on the crude level that I'm talking about, tremendous amount of sampling, okay? Okay, question, yes?
So these are the cases that are qualitatively differently?
No, I just know that the state transition graphs will be different. The Morse graphs might be the same. And typically, there are not that many Morse graphs, but I don't know a priori what's going to give me what, okay? So I'm going to compute on this, because this is where every single node is going to have to be playing a role, and every single node is playing a role. Yes?
Sorry, because I missed something else.
Yeah.
If I were to naively want to provide a set of differential equations for this, etc., you have only a few boxes in there. Presumably for each of those you have a mechanistic description. I mean,
let me give you a decay rate for each term, and for each edge, a low representation, a high representation, and a threshold. So 3 nodes, 3 parameters per edge, 1 parameter per node. I'll come back and give you an explicit example in a little bit.
But you get this number by multiplying out those things.
This number?
The parameter number.
No, I get this number by looking at possible orderings. Remember I gave you the ordering of T, P0, P1?
I see.
That's why I care so much about forgetting about numbers and just talking about order relations. Okay? Okay, so let's, but I'm doing combinatorial computations. I'm not trying to solve differential equations. So I can do a billion. That's, you know, it takes a while, but it depends on how many clusters you give me, okay? So I find a Morse graph for each one of these. If this is what I'm seeing, I assume that this is stable dynamics. So I want to go down to a minimal node in my Morse graph, and I want to see a minimal node where there's oscillation involving all terms. That's about 2% of these, okay? Okay, so this model can produce a cell cycle. But a submodel of this is already in the standard books for what a cell cycle for yeast looks like. Let's go and knock out CLIB2. And then that is what induces endocycling. And here's what you see from the endocycling. So if I knock this out, we assume that the parameters, relating to the other pieces don't get changed, because I'm looking at a sub-portion of it. And I run the same numerics now for this subgraph, and I ask, can I see oscillation in the order with maxes and mins for some? And the answer is yes. For 0.6% of the initial essential parameter values, you get a fit to this data, and you get a fit to the wild type data. And so our what? Yeah, okay. But I think there's 2 points going on here. One is biology. It's clear that we evolve to networks with particular parameters if we want to see something coming out. Change parameters and something different is going to happen, all right? So it shouldn't work for all parameter values. Second, 1 million out of a billion or a trillion, however we're counting it, that's still maybe a lot, but it's biology. It's got to be robust with respect to perturbations and noise. And so to me, this is a statement, you don't have to get the parameters exactly right to see the kind of phenomena you want. So I actually like this number. Okay, whether it's 0.6 or not, I don't care. But it should be something small, but it can't be close to 0, or else I would start worrying about the model being too delicate. Other questions? Okay, so I solved the biology problem in the sense that this model is consistent with the data. Whether it's the right model or not is, again, beyond my pay grade. Okay? But as a mathematician, I've done what I can. Yeah.
So if you knocked out other nodes, do you then compress this further and get down to?
Yeah, we'd have to do more experiments, and that's definitely not in my ballpark. Okay. Yes.
So if I understand correctly, this is like you are constructing sort of a circuit or something, like it has like gates and then this. So instead of using ODEs, there's like some pseudo current, let's call it like this, and that is giving you a result or something.
Yes. So I'm in between the Boolean, and we actually understand exactly how Boolean models fit into this, and somewhere between ODEs. We still have something that I think is computable, but I also, and I'll get back to this in a moment, I claim that if you start fitting reasonable ODEs to this, I've already told you what the dynamics is going to be. And that's the next part of the talk, okay?
So that is not, you see, it's not reducible to Boolean.
The Boolean networks, the Boolean networks are much, much smaller than what we're looking at. And I know, I mean, we're coming out with a paper, which again, I had hoped to finish by this week. This should be out soon. This other paper should be out soon that shows exactly how Boolean fits into.
So does a Boolean model predict the same?
We didn't test it with the Boolean model. But
Can you do synchronous Boolean or asynchronous?
Asynchronous. What I know is that asynchronous Boolean, we know exactly which parameter nodes correspond to asynchronous Boolean modeling. And it's a very, very small subportion of what we're seeing. And we have examples where you get the wrong set of stable fixed points, for example. Okay? Yeah, right. I'm just saying that the Boolean fits into what we're doing. And in fact, if the computation was even larger, a natural thing to do would be first do a Boolean computation, find out where in parameter space you might see something, and then search in neighborhoods of that. Right? So it's, again, I think it's important that it fit to the Boolean, because the Boolean really is, from a computational point of view, and giving intuition is really important. And so I'm very happy that we know exactly how it fits in. Does that answer?
Yeah, no, because it's very controversial. Yeah. In the case of the flight development problem, right? 15 years ago, there was a huge controversy about this Boolean model.
Okay. Yeah. Okay. All right. So let's go back to math. Okay. There's something called the Conley index. It's a topological generalization of the Morse index. It allows me to go from pure algebraic topology to statements about dynamics. Important fact for this talk is that it's computable from this state transition graph I've made, modulo choosing the right conditions on this. And this is what I'm skipping. And this is about 50 pages of our 200-page book. It's making sure that you've given rules for how to build this multi-valued map so that you can do the algebraic topology. And we have multiple rules depending on how fine we want the information, because the more rules you put in, the more expensive the computations are going to be. Yes?
So that 51 trillion number you showed, is that the number of such state transition graphs, or?
Yes, it's the number of state transition graphs.
Okay.
Yes. Okay, so for this example, if you run it through the machinery, well, I already told you what the Morse graph is, and now I'm telling you the Conley index of this is the Conley index of a stable fixed point. Conley index of this is that of a stable fixed point. Conley index of a saddle point. So I have kind of an algebraic topological language that means I don't have to look at these pictures, and I don't have to look at exactly how the arrows are going. I can understand kind of qualitatively what the dynamics looks like. Is that okay? That's kind of all I want out of that. If I were working with an ODE, then I could tell you as a theorem, modulo the geometrization, that you'd have 3 non-trivial invariant sets, each of which contains a fixed point, and that this is a separatrix. So there are theorems that let me now go from this combinatorial setting into true dynamics. I'll come back. Computation requires particular subcomplexes. You go and look at forward invariant sets, down sets from the topological sort. You now need to move up to the full cell complex. So if it's a high dimensional complex, this starts becoming expensive, but we've actually been able to do computations with 12-dimensional systems. So, you know, if we can get this morning's talk, I think Smita was talking about getting down to 10 dimensions. I'm okay with that. That's the kind of stuff, in principle, we can handle. And what's the Conley index? You find two of these sets up here, and then you just compute the relative homology. And there's code that does this, all right? So if you don't know what homology is, that doesn't matter. There are buttons to push. Geometrization. Up until this point, everything has been homological algebra. It's just combinatorics with a little bit of linear algebra. I need to make it appear as something. So algebraic topologists know how to do this. They start with a topological space. They say, oh, make it into a CW complex and use the CW complex to compute the homology. I'm going backwards. I'm saying, here's a cell complex, turn it into a CW complex. That's what I'm saying right here. I got to add the dynamics to it. What I want is that the vector field for my differential equation should have the property that if I look at any one of these forward invariant sets, the vector field on a boundary is transverse and points inward. And if I have that condition satisfied, then I'm in good shape, okay? I have actually solved the differential equation for you. Okay, so here's an example. Two-node network, so I can show you a picture. Here's the abstract cubical complex. There are two edges now, because two vertices in each direction, because there's two edges for each one of these. But this is the input to build the parameter space. And now, of course, the state transition graph is made on the blow-up complex. If I gave you this wall labeling, and I used this particular state transition graph that we make, this is the state transition graph that you're looking at. You run it through the topological sort algorithms, you end up with this Morse graph. You compute the Conley indices, and you get these Conley indices that we already talked about. Okay? Now, this is still all combinatorial. Let's geometrize. So this is for Eduardo. Yes, get your attention. Let's put Hill functions in there. That's a reasonable choice of differential equations. There are lots and lots of parameters. The only parameter I haven't talked to you about is how steep I'm going to make the Hill function. I want nice pictures, so we made it equal to 20, okay? But it's not biologically realistic, I know, but it makes the picture very easy. I would have to, it's a lot of work if I don't make it 20, okay? Notice that this cell here, and of course the color coding doesn't match, this cell here did not get matched to a nice little square. It got sent to something like this, and we kind of have formulas that tell us how to send it, okay?
So you have formulas that tell you? You have formulas that tell you what? How
to take this cell and transform it into this object right there. Not for the Hill function, but for for simpler nonlinearities, but yes.
So what you're doing is you're given the multifunction that tells you you could go to any of those, but you lose information on it. Maybe there's only a little bit of probability going to one and not to the other, but that's lost, right? It's just a non-deterministic automaton.
Right. Yes. But now the algebraic topology that I had before is valid for this differential equation. So the way I'd like to think about this is, you know, in the 80s, and we teach this, right? How do you solve an ODE, a two-dimensional ODE? You do a phase plane diagram. This is a phase plane diagram. We've automated it so it applies to n-dimensional systems.
Right. But you're overestimating.
Oh, of course. There's, right? I mean, it's only at this point that I put a differential equation in. And what I'm saying is I've already solved it. There's no need to do any more numerical computations, okay? I lied a little bit, but let's give me that.
Also here you can have like boxes that are cycling, right?
Yes. Yeah, we could, and not for this model, but for yes, right. Okay. Herbie had to leave. He already told me he was going to leave. And that means I'm getting late. Am I? I have 5 more minutes? Okay, I'll be. 5 minutes over time. Okay, I'll finish real quick. I haven't written down differential equations. Herbie was right. If I put differential equations in, I could see really complicated dynamics, right? So let's go back to where I was talking about hysteresis. So, I've kind of drawn a picture. These are, the state transition graph arrows. Put a differential equation into that cell complex. Think of it as an n-dimensional problem. What am I going to get? I could get a mess, right? The algebraic topology tells me there's got to be something in here. There's got to be something here. Well, there's got to be something here, but there could be a lot more stuff. I think that you were alluding to that. Theorem. And this is by Akash, postdoc, Parikh. You got this complicated picture because you picked a nonlinearity. I claim that you can homotope that nonlinearity to one where you see exactly the hysteresis curve without changing any of this other data. Okay? Now, if you want to go back and choose a model that looks like this, that's your problem, unless you have more data that tells me that you really care about that complicated dynamics. I'm telling you up to homotopy, because nobody knows the nonlinearities correctly, you're perfectly fine saying that that's the dynamics that's going on. Okay? Open question, is this true in general? All right. Now, I put a dot, dot, dot here. I know this is a theorem for n equal 5 and above, 1 and 2, 3 and 4, I'm not sure yet because we're talking about the pseudo-isotopy theorem, and that's not something I'm really very comfortable with, and we're trying to understand that, okay? Okay, finally, let me finish with data-driven dynamics. This is brand new. I got the results I'll show you were emailed to me, okay? So that shows you how new they are. I'm hoping to go in this direction. This is completely unrealistic what I'm doing, but if you give me the differential equation and you sample somehow the vector field, but of course I don't want to know what the differential equation is, can I figure out which region in parameter space that dynamics came from? Because if I know that, then I know what the solution to the differential equation is, even though I don't know the differential equation. So we have a theorem, modular technicalities, that says, if you give me the network and you give me a parameter node in that network, I know how many samples I should take if I'm taking a uniform sampling of the vector space. That's not particularly interesting, except it kind of gives me a baseline to start doing experiments. So here's a network with 648 parameter regions. For each parameter region, pick a parameter value. We now know how many samples we should take if we expected to get the right answer, and know that we would get the right answer if we had a nice even spacing, uniform spacing. So now let's randomly sample, but okay, I'm going to pick ODEs with S equals 20, again, to make sure life is simple for me. I'm going to use a uniform random sampling, but with 4 times as many points as I would need if I had everything perfectly aligned. And I'm going to do this 100 times. So for each region in parameter space, I'm going to get 100 samples, and I'm going to ask how often, when I run our algorithm, do I get the right region? And that's the plot. So this is 75%. So we're always better than 75%. Here's 80%. We only miss it once. And typically, we're doing better than 95%. Another way to measure this is most things that I think as to 0th order you care about is stable stuff. And there's only 2 places where we got the wrong number for the stable stuff. Okay? There's a lot of work to be done. Uniform random sampling, you're not going to get that from, say, RNA vector field data. We need to think about what happens if we're sampling from trajectories. But maybe there's something that can be done here. Okay, and I went over. I apologize. Yes, there's a lot of people working on this. Okay, thanks.